Hi everyone, good evening. Good evening everyone. Welcome to another free session on YouTube, right? Today our main segment would be to revise the microbiology segment, right? We'll be dealing with the previous year questions of microbiology. So just check whether the audio and video is clear to everyone or not, and then we'll start with the class accordingly. Yes. I'm just forwarding all the segments to everyone. Yeah, let's begin. Right, just check whether the audio and video is clear to you or not, and then we'll begin with the class. Right, let's begin. So we have our rankers batch ongoing for your June 2026 examination. In addition to that, we have our GATE XL content also available, be it your ebooks for the entire content, be it your revision capsules or your mock test series. So we have all of these things available for you. In addition, you can contact us on this number for any kind of help, for any kind of query, and our rankers batch was started on 5th of January. So whatever is your requirement, and we have various other courses as well for you to decide or to prepare for the other examinations, right? So let's begin with the class. In addition, we also have our own Lakshya batch available, which is a completely free of cost segment, which is specifically for your day-to-day practice, right? Every single day you get a task, you get free test and every single thing. So if you wish, you can join in the free Lakshya batch as well, right? So you can download our application and get enrolled in all of these things. All the links are in the description box. So let's begin. So we'll start with the microbiology segment and the recent paper, the June, the 2025 paper, right? So we'll start with the microbiology questions, and for the next 5 days microbiology will be taken into consideration from 2025 till every single, like I will try to do it till 2015, but let's see till which year we can solve it. So first question for you all: which of the following metabolites is associated with bacterial stringent response? Question is asking you which of the following metabolites is associated with bacterial stringent response. So you have cyclic di-GMP, guanosine tetraphosphate, cyclic AMP and cyclic GMP. Give it a try to the question everyone, then we'll do it together. Yes. Which of the following metabolites is associated with bacterial stringent response? This is a factual question. Cyclic di-GMP, guanosine tetraphosphate, cyclic AMP and cyclic GMP. What will be the answer? Mark the answer for the question everyone. let's see. So what should be the answer for the question? So it is guanosine tetraphosphate, right? So the bacterial stringent response, it's a survival mechanism that is triggered by the nutrient deprivation, specifically your amino acid starvation that we talk about, right? So signaling molecules involved, which of the two signaling molecules are involved? You have guanosine tetraphosphate and you have guanosine pentaphosphate, right? So together these are called as your ANS. So these are associated mainly with RELA and SOPT proteins in the bacteria. And what all things does it cause, or what all various segments does it cause? It causes decrease in the rRNA synthesis. It causes difference in the tRNA synthesis, decrease in the DNA replication, and increase in your amino acid biosynthesis genes that we have. So overall the shift is from the growth mode to the survival mode, right? So from the growth mode to the survival mode we have the shift happening in, because this is triggered during what? When do we get to see the stringent response happening in? We get to see the stringent response when we have the nutrient deprivation associated to the bacteria, when the bacteria is not getting adequate amount of nutrition. So B is the correct answer for this particular question, right? Easy, simple, right? One more question. India is aiming to be free of tuberculosis by 2025. One of the key approaches for this program is DOTS. What is the meaning, or what is the full form of DOTS? That's what the question is. Try it out, everyone. Good evening, Neetu. Which of the following is the full form of DOTS? So there's a program ongoing which because of which, or according to which, India is aiming for being free from tuberculosis. So which is the key approach, or what is the full form of DOTS? Directly observed therapy short course, directly observed tuberculosis short course, district operated therapy system, and directly operated therapy short course. So what should be the answer? Very good. B, A is the correct answer for this. Directly observed therapy short course, not tuberculosis short course. The GATE has given you, or they have given you this particular segment specifically to confuse you and specifically to what do I call, make you like mark this answer only, right? Because जैसे ही हम tuberculosis word देखेंगे, we would understand कि tuberculosis से related तो सिर्फ एक option है. So we are bound to mark the same answer. So it's a trick that is there. Okay. So remember, A is the correct answer. Directly observed therapy short course, DOTS, right? So what is the key idea in this entire segment? The key idea in this entire segment is that a healthcare worker directly observes the patient taking tuberculosis medicine to ensure the compliance and prevent the drug resistance, right? So it is a entire scheme has been initiated by WHO, which is World Health Organization, right? Next question. This question is, correctly match the bacterial type in column A with their corresponding environmental niche in column B. Column A may we have the various types of bacteria, and in the column B we have the corresponding environmental niches of the bacteria. So we need to find out the correct match for the two, right? So psychrophils, barophils, mesophils and halophils. I think two of them you can very easily correlate and mark it. So try the question everyone, then we'll do it together. And if you wish to join in on regular basis, please make sure every day 4 p.m. we'll be having a class for microbiology, because after biochemistry, microbiology is the most commonly selected topics by majority of the students. So I thought of like taking up microbiology in the segment. Last week we discussed biochemistry. So correctly match the following in terms of the type of bacteria and their environmental niches. Like where do we get to see this bacteria as being there? In which environmental conditions do you find them? Mark the answer quickly, then we'll do it together. So halophils. Halophils is very, very commonly in terms of your salt concentration being very high, right? So S being marked with fifth. Three options are there. I can eliminate this. Psychrophils is somewhere related to when I have a temperature being very less, right? So you have psychrophils related to the temperature below 15 degree. Let's see if I can mark my answer accordingly. 1 with 3, S with 5. Is A the correct answer? Yes, A is the correct answer. See, with just two of my options, I have reached to my correct answer. Then you have barophils and then you have mesophils. So barophils is like correlated to the barometer, right? Barometer is in which the pressure is calculated. So pressure greater than 380 atmosphere. And then you have mesophils. It is the temperature when the temperature is like moderate from 15 to 45 degrees Celsius. So what will be the answer? A will be the correct answer. These are all the recent year paper. GATE examination is not very difficult as compared to CSIR. Okay, if you have your factual understanding clear, if you have your facts very much on tips, you can easily get a good mark in GATE examination, right? Is the question clear to everyone? Yes? Let's discuss the next question. Okay, another question. Robert Koch used meat infused nutrient medium for which of the following purpose? So he was working on an experimentation. So it is asking you that it is used as a meat infused nutrient media for which purpose? To grow the disease causing microorganism, to demonstrate the presence of microorganism in air, to test the efficiency of sterilization approaches, and to demonstrate antimicrobial activity of the soil isolates. What's the answer for this one? Try it out, everyone. Mark the answer. Robert Koch used a meat infused nutrient media for which of the following purposes? To grow the disease causing microorganisms, then to demonstrate the presence of microorganisms in air, to test the efficiency of sterilization approaches, to demonstrate the antimicrobial activity of the soil isolate. So what should be the answer for the same? The answer for this is to grow the disease causing microorganisms, right? So he used it in one of the earliest cultures in order to grow and isolate the pathogenic microorganisms. This helped him to study the pure cultures, to establish the postulates that he put forward, and to also demonstrate the causative agents of the disease that were there, right? So what should be the answer for the same? Answer is option number 1. See, the question is very baseline, right? This is something when you study or when you initiate with your microbiology questions. So these are the very initial segments that you study. So you can see questions are getting asked from the very basic part as well. One more question. Let's try this question first. So a bacterium obtain energy from a chemical source by the oxidation of reduced NO2 minus CO2 with CO2 as a principal carbon source. So, which of the following nutrient groups does this bacterium belong to? Try it out. The bacterium obtains energy from chemical source of oxidation. From a chemical source by the oxidation of reduced NO2 minus with CO2 as a principal carbon source. Which of the following nutrient groups does this bacteria belong to? Photoautotroph, photoheterotroph, chemoautotroph and chemoheterotroph. Mark the answer for the question everyone, then we'll do it together. Done? A bacterium obtains the energy from a chemical source by the oxidation of reduced NO2 with your CO2, right? These two things are getting reduced in together. So, oxidation of these reduced formats. So, which of the following nutrient groups does this bacteria belong to? To which group is this bacteria belonging to? So, understand this particular thing. So, the question is saying that it is getting an energy from a chemical source, right? So, when the chemical source is being used, obviously it is not a photoautotroph, right? It is either a chemoautotroph or a chemoheterotroph that we talk about, right? Now, in addition to being a chemoautotroph, in addition to being a chemotroph, is the content or is the organism using its own functionality, or is it working upon something else? It is using its own caliber to get the entire work done. So, it is a chemoautotroph that we talk about. It is not a chemoheterotroph. So, some of the the example is chemoautotroph and it is using CO2 as a carbon source. So, chemical because the chemical source is being used, and CO2 because it is using autotroph because it is using carbon dioxide as a carbon source, right? So, typical examples include the nitrifying bacteria, sulfur oxidizers and the hydrogen oxidizers. So, what is the correct answer? Chemoautotroph is the correct answer for this particular question. C is the answer for this. Let's try one more. Okay, let's try out this one. Which of the following is or are the locations for ATP generation through oxidative phosphorylation in E. coli? So, this is a multiple select question, that is one or more than one is the correct answer. Where do you get to see the ATP generation occurring in E. coli via oxidative phosphorylation? Try the question everyone. Done. Which of the following sites is or are the location of the ATP generation through oxidative phosphorylation in the E. coli? Mark the answer for this. Inner membrane only, outer membrane only, both outer and inner membrane, and mesosome. Try it out. Done. Where do we get to see the ATP generation occurring in? Very simple, it is the inner membrane only. So answer for this is inner membrane. No mesosome, no outer and inner both, right? So inner membrane only. Why? Because your electron transport chain operates in the inner membrane, right? So in the E. coli, the electron transport chain and ATP synthase are located in the inner membrane, and as a result this is the main site of it. So what should be the answer for the same? A is the correct answer for this particular question, right? One more question. Which of the following, or you have to correctly match down the viruses and the nature of their corresponding genetic materials that we have. So bacteriophage lambda, bacteriophage M13, coronavirus and reovirus, we need to match it with the specific nature of their corresponding genetic material that we have. Try it out. Done. You have the various bacteriophages, like various different type of viruses and their nature of the corresponding genetic material, right? So you need to find out that what is the correct nature of the same. So your bacteriophage lambda is a double stranded DNA. Then you have the bacteriophage M13 as single stranded DNA, coronavirus as a single stranded RNA, and reovirus as a double stranded RNA. So C is the correct answer for this particular question, right? So what should be the answer for this? C is the correct answer. It's a completely fact based question, right? Easy. Done? Understood till here, right? Okay, let's do one more. So you are given the various structures in the bacterial cells and their corresponding functions, right? So structures and their corresponding functions are given to you. You need to find out that which of the following structure of the bacterial cell is corresponding to the same. So you have the cell wall, fimbriae, flagella and pili, and on the other side you have their various corresponding functions that we have. So Cell wall, what's the function? Fimbriae, what's the function? Flagella, what is the function? And you have the pili, what's the function for the same? So try the question, everyone. Right, so what will be the function of your pili? It is transfer of genetic material, right? Flagella is majorly motility. Cell wall is majorly your protection from the osmotic stress, and fimbriae is attachment to surface. So there is entirely like it is given in exact same order itself. So A, B, C, D, like P1, Q2, R3, S4, P1, Q2, R3, S4. So A is the correct answer. No change, no alteration, same. A is the correct answer for this particular question. Understood, everyone? Yes. Let's do one more question, last question for today. Which of the following statement regarding the microorganism is or are correct? Which of the following statement regarding the microorganism is or are correct? The free living bacterium is a human parasite. Then you have Myxococcus are a group of predatory parasite. Dictyostelium is a slime mold that aggregates to form the social groups, and then actinomycetes in the soil are involved in producing the earthly odors. Mark the answer for the question. Last question for you today. Done? Which of the following statement regarding the microorganism is or are correct? So, free living bacterium, Wolbachia as a human parasite, Myxococcus are a group of predatory bacteria. Dictyostelium is a slime mold that aggregates to form the social groups. Actinomycetes in soil are involved in producing earthly orders. So B, C, D, all three of these are correct. And then first option is wrong because it is not a human parasite. It is not free living. It is an obligate intracellular symbiont. It infects the arthropods and nematodes and not humans, right? So it is incorrect. First is a wrong statement. Other three statements are correct, right? So B, C, D are the correct statements for the same. Understood, everyone? Yes. Right, so these were the questions. We have various other questions. We'll discuss these questions in the next class. I hope till here everything is clear to everyone. I will be conducting a session tomorrow as well, 4 p.m. same timing. So try joining in at the same time, and we'll discuss the further segments. Okay, so all the best. Good luck. We have our own GATE XL capsule revision capsules available for you for revising, and practicing your last minute segment, right? So all the best. Let's meet in the next class.